There is work out there by James McGaw, who's at a university in California, who studies people with a condition, although it really shouldn't be called a condition, called superior autobiographical memory. And these people can essentially remember a huge amount of detail about every day in their life.
Here we are in the heart of Edinburgh Neuroscience's offices, and we have Anna, who's preparing the weekly seminars email for you all, being created first thing on Monday morning, as always.
You're just going to get a video of me laughing. That's all it's going to be.
Hi, I'm Tara Spires-Jones in the Centre for Cognitive and Neural Systems.
Hi, my name is Duncan Martin. I work in the College of Medicine of the University of Edinburgh as the Business Manager for Imaging.
My name is Alexa Morcom, I'm from psychology.
Hello, my name is Grant Mayer, I'm a neuroradiologist.
Hi, my name is Matt and I'm a neuroscience PhD student here, working on multiple sclerosis.
Hello folks, this is Ian Deary from Edinburgh in the Lothian birth cohorts with three of my colleagues.
Riccardo Marioni, David Hill.
Gail Davis.
Neuroscience is so massive, that's the thing. You can look at it from so many different perspectives that I think there's something for everyone.
I love that it's all actually new, new to the world.
Each of the little bumps is a different amino acid, and this one that's coloured in orange on its own, when it's mutated, is enough to cause severe epilepsy and autism and intellectual disability.
So I have brain sections on slides. This is my box of slides here. I do this quite a lot. This is a big part of my PhD.
My favourite part of the work? I really miss lab work. I haven't done any for a long time, and that would have been my favourite if I was still doing it, but I think seeing students get good results and be excited and inspiring a new generation.
Most of it's just drudgery though, isn't it? Doing the same thing, keep going, try and work out why it's not working, keep going.
That's why it's so exciting when it works.
Yes, it is.
And seeing Kathy's face light up when it does work. Coffee is also necessary all day. We have cute little IGM cups now,
unfortunately. I love that I get to use my brain, I get to think, I get paid to think. I think that's the best job in the world, and then I get to do experiments based on my thinking, and sometimes they work.
I think one of the most amazing time points during brain development and aging is actually the postnatal development, because there are billions of brain cells and they all need to get to the right exact spot. They need to grow the exact branches, and most importantly, they need to make the right connections in order to be a functional brain. And if you think about how amazing that is, and there's so many things that can go wrong, it is a miracle how often it goes right.
My name's Sue Fletcher-Watson. I'm a researcher at Edinburgh University in the Patrick Wild Centre. I'm going to do a bit of filming of a big international conference that I'm at to help you give you an idea of what it's like at one of these big meetings. So first of all, I'm just going to show you the main room where the keynotes are so you can see the scale of it.
So on average, and everyone here should be above average, but on average, the brain contains about 100 billion neurons.
The question we're interested in is why is memory selective? We use this technique called optogenetics. That's a revolutionary technique that all revolves around small proteins that are found in some various microorganisms that are sensitive to light. Now, by using some genetic tricks, we can express these proteins in neurons, and then the magic happens when you shine the light onto these neurons, and you can either inhibit or activate them depending on the light-activated protein you use. That's why we can see this beautiful, pretty much silence of neural activity when the light is on. That changes into this symphony of neural spikes as soon as you turn the light off.
The technique that I'm doing at the moment is very new, and it's very cool.
My goal is to understand how neural circuits function, how they process information in a living animal. I decided to work on a very simple brain, and that's the nematode C. elegans or Cinorhabditis elegans. You can trace back the lineage of the whole community back to one person, Sidney Brenner, who had this idea of establishing C. elegans as a model for understanding biology.
At Milestone House, we try and pick up where hospitals seem to stall a bit in the rehabilitation of patients with alcoholic-related brain damage, try and expedite their safe discharge. So we try and get people better in a safer way, where they get insight into their problems and all that in an environment where they're cared for and loved.
Alright, on Fridays the Becker group has lunch together and one of us gives talk about her results. Today that's Hannah, who's waving there in the back.
I find the brain to be amazing because of its output. It produces art, produces literature, music, science even, philosophy, and how it does that, I think, is based on its complexity.
Favourite lobe of the brain? I think it has to be the frontal lobe. If you're a neurologist, you've heard of this famous guy called Phineas Gage, who was packing dynamite and got a rod blown through his brain and then took up through swimming. You can't beat that.
My favourite lobe of the brain is the frontal lobe because it's responsible for being in charge.
Oh, I think it would have to be the frontal lobe.
I'm a frontal guy.
Well, mine has to be frontal lobes.
Definitely the medial temporal lobe, and most importantly the hippocampus, which is at the seat of the medial temporal lobe, which is the interface and all the machinery that is responsible for automatic recording of all the experience that you encode as you walk around living your daily life.
It's not a lobe, it's a part of the brain, it's the white matter. It's the area containing all of those axons that are myelinated, that connect all the different lobes of the brain together. This is 50% of the whole human brain, and it tends to get ignored, but it's really important.
So I'm Pete Connick, I'm an academic neurologist at the Anne Rowling Clinic. I work on mainly MS, and what I'm interested in doing is two big things. One is looking for treatments for the neurodegenerative element of it, the progressive phase. We don't have any treatments for those patients. And the second is trying to use the treatments we do have better by personalising care.
So the Neurological Voice Bank project is a Ewan MacDonald Centre project based at the Anne Rowling Clinic, and this project aims to create personalised synthetic voices for use in communication aids.
Please call Stella. Ask her to bring these things with her from the store.
So when people may lose their speech due to degenerative disorders, they may need to use a communication aid, and we're hoping to provide a personalised voice that they can use in those communication aids.
So we're in our fish facility and we're using the zebrafish to investigate how the nervous system is built and how it's repaired after injury, and it's obviously very, very important. It's something that we can't do and the fish can quite brilliantly. So we're using the fish to investigate what the cellular changes and the molecular changes are that lead to successful repair both of nervous system damage and of loss of neurons that happens in us after injury, after stroke. The immediate human impact is that we do a fantastic amount of training. So we have Hannah here, for example, who's doing her PhD.
So probably my favourite part of my day is when I get to take my fish to the microscope and take a look at how their nerves have grown. It's really fascinating.
I think neuroscience currently is at a very exciting time because the field has advanced enough that we can finally ask the right questions to study. And now all we need to do is get answers to all these exciting questions.
So today what we're going to do is set up a new experiment looking at how adults of different ages use memory cues differently. The technique we're using is EEG, which is a recording of the brain's electrical activity, which we use to study the brain in action and see what's happening while people are trying to remember things. Of course, we can't see from this exactly what's going on in terms of how she's thinking, but with some hard work, some data analysis down the line, we will be able to make some comparisons which help us to understand memory better.
Hi, I'm Sue again from Edinburgh University at the International Meeting for Autism Research. So I'm just going to show you what a poster session looks like. So this is one of the poster sessions at IMFAR. So basically when you come to an academic conference, you can, if you're lucky, give a talk. But if you don't get the option to give a talk, then often you'll be offered the chance to present your research in a poster.
The most amazing of the brain is that it controls every single movement of our body.
General Assembly of the EPAD Consortium, which is the European Prevention for Alzheimer's Dementias Consortium.
Alzheimer's disease is a disease of midlife that kind of presents itself in later life. So actually, if you see someone who has Alzheimer's disease, it's likely that they'll have changes in their brain at a much earlier age, maybe like 20 years before they start to show symptoms. And that's the kind of age group that we're focusing in on now.
One of my research interests is the ability to dual task, which is to perform two tasks simultaneously. And we know that in healthy adult ageing, older adults are able to do this. However, in patients with Alzheimer's disease, despite being able to perform tasks on their own relatively well, when they have to perform them together, they perform poorly.
I love neuroscience because almost every day that I work in neuroscience, I do something different. Whether it's a new experiment, a new technique, whether it involves much more analytical prowess, what I do every day changes.
Hello, is that Mrs. Brown? Hi there, it's Chloe from the 6-day sample study. How are you today?
Well, the 6-day sample are 1,208 people born on 6 days in 1936. They did an intelligence test when they were 11 and were followed up for 16 years until they were 27. It's such an amazing wealth of data that's available on them, and we are following them up now at the age of 77. It's just amazing to be able to speak to people and meet them in person who I've only ever seen in a database and heard so much about their lives.
My age of the brain, well, I studied the brain between 70 and 90. And here's a 3D printed brain of one of the Lothian birth cohort, 1936. Actually, it's in pretty good shape. I'd be pleased if my brain looks like that when I'm in my late 70s.
Within neuroscience, we have the ability to investigate how the brain works and to understand and treat the diseases that affect it.
In 10 years' time, what I'd love to achieve is for us to be able to personalise your care prospectively. At the moment, we largely react to what happens to you. And that means we lose time and we lose the brain. And we can't afford to do that. We've got good treatments now. What we'd like to do is when we make a diagnosis, predict for you how it's going to go and then advise you better on what the treatments are. I'm hoping that's when we can be.
I think there's going to be a lot of developments hopefully in improving intonation and naturalness of the voice, because obviously the voices we're creating are still quite synthetic. So I think there's a lot of exciting developments.
The coolest thing that we could see is the artificial creation of memories.
This work we're doing on epilepsy mutations is really exciting, and it could actually change people's lives. That would be brilliant if something I've done contributes to kids getting better, you know, that'd be the best.